This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone, and welcome to another episode of the Anthrobiology Podcast. Before we dive in, I would like to take a moment to put out a call for people who research cannibalism. They don't even have to be a biological anthropologist. Historians, archaeologists, wildlife biologists, and paleontologists are all welcome. Also, if you know someone who has eaten a placenta before, I am very interested in talking to them. All you have to do is DM me on Instagram or Twitter, both at Anthrobiology, or email me at gabby.lapera@anthrobiology.com. That's G-A-B as in boy, Y.L-A-P as in Peter, E-R-A at anthrobiology.com. Turning to today's show, today we have Dr. Bartelink from CSU Chico to talk about isotope analysis. This is a great crossover episode. It's kind of like when the Wonder Twins get together in Super Friends. It's basically all like, when chemistry and biological anthropology unite, their powers are unlimited. Okay, so maybe not that, but it's pretty cool what they're using isotopes for right now. Isotopes can answer a lot of questions about migration patterns and food consumption, both in the past and in modern forensic contexts. So please buckle up for another great episode. Oh, and if you can, I would love it if you could recommend your favorite episode to someone you like. Let's spread the good word about biological anthropology together. Hello, everyone. I hope you're having a great day. I am in my apartment in San Francisco, California, and joining me over Skype is Dr. Bartelink of CSU Chico. Thank you so much for joining me.
Thank you for having me.
I have been chasing after you for months now, and so I'm very excited to be interviewing you today, especially because you have a very cool specialty, and I'll kind of let you introduce yourself. Tell us what we should know about you, Dr. Bartelink.
All right. Well, I'm a forensic anthropologist at California State University, Chico, and over the last 15 years or so, I've been really interested in stable isotope applications in both forensic anthropology and also in bioarchaeology. So applying these methods to modern criminal cases as a means of identification for law enforcement, but also looking into the past to look at prehistoric mobility and diet patterns, even things like breastfeeding and weaning patterns in prehistory. So many different applications of stable isotopes in anthropology. It's becoming a really exciting field to work in.
So there's a bunch of stuff that I want to break down in what you just said. First of all, I want listeners to note that you do both forensic and archaeological. You work in both contexts. And I think that is something that's special to anthropology, where you have a lot of anthropologists who are actively working in both current populations and archaeological populations at the same time. I can't really think of any other disciplines that necessarily do that.
Yeah, it's definitely one thing I think that makes anthropology unique. And while there are people who specialize more in one area over the other, it's not too uncommon to have people who do research in both bioarchaeology, so dealing with ancient skeletons or historic skeletons, and then also forensic anthropology, dealing with modern forensic cases for law enforcement. And so isotopes are just become kind of a new part of this still emerging fields.
Can you explain what stable isotope analysis is and why it can be applied across both fields?
Yeah, so stable isotopes have been used for well over half a century. In archaeology, the first applications came about in the late 1970s and sort of were used to track the introduction of corn agriculture into the eastern United States. That was sort of the first application in anthropology. Isotopes are atoms of the same element, so carbon-13, carbon-12 as an example, and they have the same number of protons and electrons, but they have a different number of neutrons. And so carbon-13 is heavier than carbon-12 and will go through chemical reactions at a slower rate. And these ratios are going to be different between different types of plants. For example, plants that have different photosynthetic pathways. So corn, as an example, has a different isotopic signature than, let's say, something like wheat. And so these isotopes get incorporated into our bodily tissues from the food that we eat and other isotopes from the water that we drink. And those isotope ratios can be measured in things like bones and teeth, hair, and fingernails, and they all provide different snapshots of time of a person's life history. So perhaps what they ate and also maybe where they got their food and water from, so kind of an indication of their travel history or mobility.
So you said a lot of really interesting things all at once. So let's I think the first thing that we should dive into is you mentioned that you can get isotope ratios from different body parts, so hair, nails, teeth, bones. How are the different isotopes getting into those in the first place?
So basically, when your body is building protein, it's coming from basically the dietary protein that you consume. So your tissues reflect what you eat. So you are what you eat isotopically, and there are some offsets between the food you eat and how they are incorporated into your tissues. But basically, your tissues are being built off of food and water, and different isotopes record different information. And so you could be looking at something like human hair or fingernails or even bone mineral or tooth mineral and get information about a person's drinking water from the oxygen isotopes in those tissues. Whereas other tissues like bone collagen or even the dentin, which is underneath the enamel in your teeth, or hair or fingernails, which are keratin, they all can give you information about diet as well. So they can tell you things like carbon and nitrogen isotope ratios or even sulfur isotope ratios. And so
I wanted to ask you, all of those tissues are very different from each other. And I think the thing that is most different about them is when they're forming in your life, right? So like your nails, for example, are growing all the time. Your hair's growing all the time. But your bones kind of fuse at certain ages. So does that mean that the ratios that you'd find in each of those would be different?
Yes, so they can be different for a variety of reasons. So they're recording different snapshots of time. So if we look at something like fingernails, which grow roughly like 2 to 3 millimeters a month, that's giving you basically these almost like weekly or monthly increments. You can get small sections of fingernails 2 to 3 millimeters long, and that's going to give you information about the recent diet or travel history. And the same thing with hair. Hair is growing about a centimeter a month. So you can take many small sections of hair, so we basically call this like an incremental analysis approach. And that'll give you much more detailed information about what that person was eating and where they were traveling in the weeks and months prior to their death. So you could use this to look at mummies that have preserved hair, modern people today, including forensic cases. Now, for something like teeth, they are locking in the signature at the time that the tooth formed, and typically those signatures do not change throughout your lifetime. So if you take someone's first molar that's already developing around birth, and the crown is usually completed by around age 4. So that's giving you kind of a snapshot of that person's early dietary history and residential patterns. Bone is going to be the most complicated tissue to work with because bone is constantly remodeling itself, or the bone cells are being destroyed and recreated. And so if you, let's say, moved when you were 20, your bones might take a decade or longer to sort of change towards the new water source and the new place where you move to. So bone has a slow kind of remodeling time, whereas teeth are just snapshots of childhood when they formed, and then hair and fingernails are weeks to months prior to death.
Yeah, and I think this is something that people are intuitively familiar with. I think there's that kind of common fun fact that you get a new liver every 3 to 7 months. It's the same thing with bones, just on a much slower time scale.
Definitely.
And one thing I actually wanted to say about teeth, and I was really freaked out when I first learned this, is that like you're basically born with all the teeth that you're going to have, right?
Well, you were born with essentially forming tooth buds. And so you have 2 sets of teeth. You have your deciduous or baby teeth, and those are forming in utero, just a few months in utero. And those will be pretty much erupted by around age 3. But while those teeth are fully erupted, as a infant is making that transition to a toddler, the adult teeth are continuing to form below, underneath those baby teeth. And that process goes on through adolescence. And by age 12, you pretty much have all of your adult teeth should be fully erupted and should have pushed out those baby teeth, with the exception, of course, of your third molars, your wisdom teeth, which may not come in until you're 17 to 21, and sometimes not at all because they're often impacted.
Yeah, which means stuck.
And yeah, And the baby teeth are going to be recording information starting in utero all the way into early childhood. And then your adult teeth kind of pick up where those teeth leave off with some overlap between the two sets of teeth.
I think one of the weirdest things that I've ever seen in a lab was a child's jaw where they had most of their deciduous teeth, and something had happened post-mortem where there was a hole underneath the deciduous teeth, and you could see all of the adult teeth just hanging out below
Correct.
the bone line. I'll find a picture of that and put it online for listeners, because it's really wild to think about you just walking around with two sets of teeth in your skull for a while.
Absolutely. And if you look at an X-ray of, someone who's like 5 or 6 years old, so a young child, you can see the baby teeth are fully erupted, and by age 6, the first adult molar is, but all the other teeth are still below, and they are just going to start emerging depending on what time frame they normally erupt. And it is pretty weird to see all those kind of teeth jammed into that one little space there.
Yeah. Okay, so we have all of these different body parts that we can take an isotope analysis of, and they have different, as you said, snapshots into the person's life. What are some of the things that you can determine from isotopic analysis?
So if we start with something like bone, and you're just doing what we call sort of bulk dietary analysis, this is where you would extract the collagen from the bone. You can also radiocarbon date things like bone collagen, and that'll give you an idea of when that person died. And that's using a radioisotope, so radioactive carbon-14. So that's an unstable version of carbon.
So when you say when someone died, you mean like in what millennia, like what year they died in, as opposed to when they died in their life cycle?
Correct. Yeah, it'll give you, depending on the precision of the instrument, usually within at least a century of when that person died, although we're getting a little bit better with that. So they sort of time once the date of death is pretty much just what you're trying to get at with that. So that allows you to put things in time. So if you have prehistoric skeletons that you're studying, you can figure out how old they are and see how things like diet change through time or mobility patterns change through time. But with bone, we can do the collagen, and collagen is primarily reflecting the protein that you consume. So it's heavily biased towards the protein sources of your diet. So that you could tell, for example, people are eating C3 plants, which is most of the vegetation on Earth, about 95%, versus C4 plants, which would include, usually they're cultigens, so like C4 plants include things like corn or maize, amaranth, sugarcane, millet, and sorghum. Those are the most common ones that people around the world might eat. And because the C4 plants, they have a different photosynthetic pathway. They use a 4-carbon molecule, whereas a C3 plant uses a 3-carbon molecule. They end up with higher carbon isotope ratios. And so you can tell somebody who's a corn eater from, let's say, a wheat eater based on the carbon isotope signature.
Right, because the big C3 plant that we would be most common with is wheat.
Yeah, wheat and pretty much almost all the vegetables and fruits that we eat are all C3, and there's just a handful of C4 plants. And also it's worth mentioning that just by chance, marine foods, so if you're eating, let's say, marine fish, Pacific caught salmon, marine mammals or something like that, they have values that are very high as well, so they can look similar to corn. And in a place like prehistoric California, where I've done a lot of work, we really look at that marine signature versus a terrestrial C3 signature. And so you can really get a sense of how much did marine foods contribute to the diet in a prehistoric population, which is pretty cool.
Yeah, that's amazing. I just want to give a quick explanation of, well, I'm not going to give you an explanation of photosynthesis, but I just want to remind listeners that photosynthesis is basically how plants create energy using the sun. But when you say they have different photosynthetic pathways, what you mean is they are creating energy in slightly different ways using slightly different molecules. And that's why you have the carbon 4 versus the carbon 3. So I actually have a question. Okay, so I think historically in the past, it was probably a lot easier to understand differences in diet over time. So for example, I worked with a couple of historic populations where you could see who had moved to the colonies a long time before they died because they had a much more varied carbon signature, right? So it looked like it was a kind of an attenuated signal. So you saw a lot of wheat and corn
Mhm.
in their bone, and then you could see who had just arrived because they had much more of the carbon-13. And then you could see people who had been living in the colonies their entire lives and had been eating mostly corn. And I think potentially it was a lot easier in these historic populations where there was a lot of differentiation between different sites. What about now? Is that something that still happens, or is that like really confined to historic or prehistoric times?
No, it's still true today, and there's a number of studies that have, and usually they use hair because it's easy to get hair or fingernails from living people. It's more difficult to sample from deceased individuals. But there is geographic patterning in diet, which can be tracked through isotopes in almost every country where they've looked. In the United States, we have a lot of corn and sugar products in our food. So think about high fructose corn syrup, cornstarch, things like that. So we actually look like we consume a lot of C4 in the United States. But if you were to compare us to, let's say, a population from Great Britain, for example, you'd see that they use beet sugar instead of sugarcane, and they're going to have a more C3-like signature. And then if you were to hop over to Mexico or somewhere in Central America, you'd see that they have tons of corn in their diet, and they have a much higher carbon isotope signature than U.S. Americans. So you can kind of differentiate populations on almost a regional scale based on cultural dietary patterns. And you can also look at things like socioeconomic differences using isotopes within a region. Perhaps individuals with access to better quality foods are going to have different isotopic signatures than those who have less access. And you can definitely get regional, like I'm sure there's differences between what people are eating in the South versus a place like California. And you would see some differences. If you were to go to like Chinatown in San Francisco, I imagine you would find dietary differences that look more C3-like there than maybe even in other parts of San Francisco. So you can explore a lot of interesting questions with isotopes in modern populations.
That's incredible. I had no idea. I really thought that it would be something that was more isolated to the archaeological past. This actually leads into my next question, because I know that there are some isotopes that you can use to kind of determine people's place of origin based on the water that they've been consuming and like the earth that they've been around. Can you tell me a little bit more about that?
Certainly the dietary isotopes like carbon and nitrogen, and to some degree sulfur, can provide some information about a region of origin, because if you see that really high C4 signature, it's often that the person is from Latin America. But that's just sort of general dietary patterns. If we can kind of build upon that and look at things like drinking water. So the water that you drink, we're looking at the oxygen isotope composition of that water. And most of the water that people drink in the United States is coming from the tap. And there are significant differences in tap water oxygen isotope values across the continental United States. We also have to recognize that the water that people drink is not necessarily local to that area. So San Francisco gets its water from the Hetch Hetchy Reservoir. So it doesn't actually look the same as maybe the precipitation water in San Francisco. But those isotopes are going to be essentially incorporated into your hair, your teeth, your bone mineral, and your fingernails. And they can provide information on where you got your drinking water from. Of course, the supermarket kind of thing that happens, that global supermarket where foods are coming in, bottled water, can confound some of that for applying it to today's populations. Although most bottled water is actually your local tap water that's been sold back to you after being purified. So most bottled water is too far from the actual source compared to your tap water. Something like Fiji water is definitely from Fiji. People have tested that. There's a whole area of food forensics that looks into those kinds of issues too. So really, it's a tracer of where you got your drinking water from for that particular isotope system.
So I have a question. Does that mean that there are maps available that tell you like, okay, so if you have this oxygen and strontium ratio, this is potentially where the person came from? Or how would you, because you need to compare it against something.
Correct. There are maps, and the term that gets used to describe the patterning they're called isoscapes. And isoscapes is a term that basically means isotopic landscape. So the geographic distribution of isotopic variation in the continental United States has been sampled through people collecting tap water samples, even precipitation water samples. And they put it into a giant database, basically a website called waterisotopes.org that Gabe Bowen from University of Utah helped to develop. It's a massive database, and people can get an account, they can use it. So there's really, really good sort of baseline reference data for all kinds of applications, hydrology, ecology, archaeology, forensics, where you can use that source of data. So to use the sort of isoscapes, you need to look at the variation across, let's say, the continental United States. And then you can break down that variation into sort of bins or intervals and assign each one a different color. And then basically project that onto a map using GIS. So you'll see different color patterns and bands that represent different ranges of isotope values. And so the way that they usually show, like the Rockies, where you have very low oxygen isotope values, often look blue or purple, whereas the Gulf Coast, where the oxygen isotope values are very high, you would see them as being kind of orange to red. So you could basically take a value from hair, nail, or tooth, and using an equation, convert it to the same scale that the water's on, and then project onto a map the locations that are consistent with that value. And it's not going to tell you exactly where someone was from, but it can tell you where they're not from and the possible locations where they may have come from. And hopefully you have other lines of evidence that can help narrow that down as well.
Right. And once you've narrowed it down to a particular region, there's other things that you can look on in the skeleton to help you narrow it down even further, right?
Yeah.
So like hopefully you could be like, okay, well, I know that this is a male about 6 foot tall and he's coming from the Rockies. So I will narrow my search to missing peoples from the Rockies.
Correct.
And then the other thing that I saw in your research that was really interesting was that you were using multiple isotopes at the same time to help narrow things down.
Yeah, the more isotopes you use, the more sort of discrimination between groups that you can get. So two groups might look similar in one isotope, let's say nitrogen, which often looks similar between populations, whereas maybe carbon is very different. So looking at multiple isotopes just gives you several different lines of evidence to sort of look at and might help you differentiate a little bit more clearly between groups or maybe in assigning origin of a particular individual. Something like strontium isotopes, which basically reflects the source of where you get your food from, is going to be a really good isotope if your unidentified person is from perhaps a rural area where they mostly ate locally grown foods, or they're from a developing country and didn't really have access to supermarkets. And so some isotope systems will work well for certain kinds of questions or scenarios and will not work as well for, let's say, maybe a person who grew up in San Francisco who were eating multiple foods that were coming from various places across the planet and have a value that doesn't necessarily represent anything particularly local to that environment. But it does work with helping with identifying people who crossed like the U.S.-Mexico border, which is one of the research projects that I've worked on with several of my students and colleagues. And so we've tried to use isotopes, multiple isotope approach to try to narrow down a region of origin where a person who died crossing the border might have come from. And that could help with identification and ultimately return of the remains back to a family member, let's say in Mexico or Guatemala or Honduras.
Yeah, that's incredible work that you do. I've talked to some people from I'm blanking on the name, but they help identify remains of soldiers that were missing in action.
Sure, yeah. The Defense POW MIA Accounting Command, or DPAA. So they still have the lab in Hawaii, and they have the other lab in Nebraska. And we do collaborate with the one in Hawaii. We do a lot of their stable isotope preparation work, and they have built the capacity to have their own isotope facility at their laboratory, which I had a chance to go visit last summer. It was really nice.
Yeah, it's really cool. I feel like the thing that's so cool about what you do is that you can and apparently do collaborate with so many different types of people across the discipline. And I was hoping you could maybe just tell us a little bit about that. So maybe something about your forensic work and your archaeological work, kind of whatever stories stand out to you.
Absolutely. So if we look at my archaeological work, which is, I'm going to call it bioarchaeology because it's sort of looking at archaeology through the lens of the skeleton and trying to reconstruct what people's lives were like in the past. And so I've had collaborations with colleagues at UC Davis, at San Francisco State, San Jose State University, and many other institutions where we've looked at things like prehistoric diet in the San Francisco Bay Area, and we've had actually a lot of support from the Ohlone Indian tribe in much of this research. And so we have looked at hundreds of skeletons. We've been able to reconstruct diet of these individuals. We know that people that lived in the Berkeley and Emeryville and Richmond area were eating a lot of marine fish, a lot of high trophic level foods from the bay. And when you move down the East Bay Shore towards Fremont and towards the Santa Clara Valley, all of a sudden people's signatures, their sort of isotope signatures, start to transform into more terrestrial-like signatures. And so you can get a sense of how much people were using the bay in prehistory for their food versus terrestrial resources. And it sort of varies very significantly based on geographic location, which is very cool. Some of our other studies, we have looked at evidence of salmon consumption in interior Central California and Northern California. So salmon are growing up and developing in the ocean, swimming into the Central Valley through the Sacramento River. And even in a place like Redding, so very far from the coast, you can see evidence that people were consuming salmon because it has a marine signature, and you can estimate how much of their diet was actually coming from salmon in prehistory. And so these are really interesting questions that would be almost impossible to really fully address without using something like isotope analysis. You can certainly look at animal bones and other lines of evidence, but you can never really get at how important was a particular food source to a person's diet without using something like isotope analysis.
Definitely. And having done zooarchaeology in the past where we dig through the midden, which is archaeology talk for the trash, and counting animal bones and like trying to reconstruct diet from that, it's hard. It's hard work. And I feel, and sometimes it's hard to separate like what is refuse, right? Like what is just what's a commensal animal? What's something that people actually eat versus just like a dog that died in the trash heap? And I feel like your work works great with that kind of zooarchaeological and historical research to kind of create a fuller picture of the past.
Yeah, and there's just so many questions that you can address with it. Working with colleagues at UC Davis, so Dr. Yelmer Erkins, and then his former PhD student of his, Dr. Alex Greenwald, who's now at the University of Utah. And I've collaborated with them. They did the majority of the work, but they were really interested in reconstructing breastfeeding patterns and weaning patterns in prehistory. And so if you take something like a first molar tooth and you actually thin section the tooth dentin, which actually has both collagen and mineral in it, you can actually reconstruct when the baby is essentially breastfeeding, because the nitrogen isotope values will start to increase because the baby is sort of like one trophic level above its mother, the same way that a human eating a deer would be one trophic level above the deer.
Right. And trophic level, I think it's kind of intuitive from what you said, but just in case, it's when you look at a food web, which you might remember from freshman year biology, which is that web that has the grass at the bottom and then maybe like a cow eating the grass and then a wolf eating the cow. Each one of those is a trophic level. So the grass is a trophic level, the cow is a trophic level, the wolf is a trophic level.
And so with the trophic level, the values are usually somewhere around 2 to 4. The term we use is per mil, which is sort of like a percent, but it means parts per thousand relative to a standard. And that's just how isotope data is reported. Mm-hmm. But, a baby would be 3 per mil above its mother while breastfeeding. And by looking at the timing of when these different sections of the dentin were laid down, which we can estimate from tooth growth patterns, you can kind of determine at what point in time, what age the infant or child was when they were weaned. And so you get this sort of increase in nitrogen and then a drop, and then it often will start to converge with that person's adult bone value. And that's really interesting because you're looking at people who died as adults, but their breastfeeding and weaning record is being basically held in their first molar tooth. We're looking at the adult molar here. It's going to come in at about age 6, but it'll be forming from birth to around age 4. And we're looking at that laying down of the dentin during the tooth growth. And the nitrogen isotopes are providing that record of the infant diet and then moving into the childhood diet. You can even work your way into teeth that form later in time, like a second molar or third molar, and figure out what people were eating throughout their late childhood through adolescence. And so you can get kind of a lot of life history information about people's childhood, even from the adult molar teeth of adults.
That is really cool. And I think that the question of when to wean is something that people are still concerned with and are still studying. And the truth is that there's a lot of variations amongst different cultures. So I think that, I don't know, I think this is so interesting, and I'm struggling to think of something more interesting to say than that about it.
And it's not uncommon in some of these Native American societies and other traditional societies that weaning may not actually take place until sometimes after age 2 or 3. So that's not too uncommon, although that would be strange for people here in the United States to hear today.
Yeah. Yeah, it's really interesting. It's definitely a window into another way of being, and potentially a place that we'll return to one day. Who knows? Can you talk a little bit about some of the forensic uses that you've seen with stable isotope analysis?
Yeah, so the larger scale projects, and I mentioned earlier the border crossers. So we've been working with Texas State University and Dr. Kate Spradley and her team there to try to help with determining region of origin of people who died in Brooks County, Texas. They died around checkpoint in a town called Falfurrias. And every year there's sometimes as many as 200 deaths from people trying to walk around the checkpoint and they get lost, and their bodies are often not found for long periods of time. Even though they're roughly, I think, 80 miles inside of Texas, they're actually dying just trying to circumvent this checkpoint. And it's become a huge problem for identification purposes. And so we've been working with Texas State and then more recently with the Pima County Office of Medical Examiner in Pima County, Arizona, Tucson, with Dr. Bruce Anderson and Dr. Jen Volner. They also have hundreds of individuals who die every year along the border that they're trying to identify. And so we've been trying to apply these methods to try to repatriate these remains back to the families. And so the isotope data can just provide some clues that can help our colleagues with making those determinations. And so we're still at a very early stage. We do have some crude isoscapes for Mexico, but a lot more data needs to be collected. But we're hoping that eventually this will allow us to narrow down at least country of origin and maybe even region within a country for Latin America. And that it would be a very useful tool for repatriation and for moving the identification along. So that's a large project that we've been working with. We've also been collaborating with the Department of Defense, the DPAA laboratory in Hawaii, and they're interested in identifying U.S. service personnel who died in past wars and conflicts like Vietnam, the Korean War, and World War I and World War II. The isotope data can help with determining whether or not a person was a local, like an indigenous person to, let's say, Cambodia or Vietnam, versus a soldier. And so we're looking at isotopes that might help us pick the soldiers out from those who are local individuals. Sometimes they have small fragments of bones and teeth, and it's very difficult for them to make a determination of whether they actually have soldiers' remains. So this is large-scale applications of isotopes, and then on a smaller scale, we're using it on just individual forensic cases. So if Sheriff Coroner's Office brings a case to us, we do a full forensic analysis of the skeleton. We provide information on the person's sex, height, ancestry, and age at death, and any trauma that might be present. But sometimes even despite our best efforts and the best efforts of the investigators, they cannot identify the person. And in some of these cases, we've been able to do stable isotope analysis, and maybe it'll help with determining if that person was a local or non-local to the area, or perhaps even somebody from another country.
That's incredible. I mean, especially the forensic research is heartbreaking, but it must feel very good to be able to contribute so tangibly to solving these different cases.
Absolutely.
Okay, well, is there anything else you would like to say about stable isotope analysis or anthropology in general?
I think I'd like to say a few things. I think it's really important for people going into forensic anthropology and bioarchaeology to sort of learn new methods. And so stable isotope analysis was not something I sought out initially. It was something that my PhD advisor at Texas A&M University did. And I did not like chemistry until I took her classes. And so I was actually a PhD student when I got interested in isotopes and had no plans to go into this as with any kind of expertise. But over time, I just started to see the value of it. And I think it really can help answer a lot of really interesting anthropological questions, both forensically and also in a archaeological or bioarchaeological context. It's an exciting time to be involved with chemistry and anthropology, and it's certainly an area that's attracted a lot of females to the field, which is something that we desperately need, as well as other diverse communities. So definitely a really emerging area within anthropology that has a lot of potential.
Yeah, it sounds like it. I think I harp on this in every episode, but I feel like anthropology is one of the few disciplines where you can do literally almost anything.
Yes.
And it's still relevant.
Absolutely.
Yeah, because we're trying to understand humanity, and that's a big mission.
Absolutely.
Anything else you want to say?
Not that I can think of.
Okay. No, that's totally fine. I have 3 questions that I use to close out every single interview.
Absolutely.
So if you are ready,
sure.
I will ask them. Okay, the first question is, if someone wanted to learn more about your work, where would be a good place for them to start? Is there an article or a book or something else that they could take a look at to understand more about what you do?
Certainly some of my recent publications, like I have a paper that I published with Leslie Chesson, one of my colleagues, and it's freely available. It's from a new journal called Forensic Sciences Research, and it's actually published out of China, but it's a high-quality forensic science journal, and all of the articles are open access.
So I read that entire paper, the one that you're referencing. I have it in front of me right now. It is Recent Applications of Isotope Analysis to Forensic Anthropology. And it was so great. I highly recommend it to anyone who wants to know more about your work. If you hadn't said anything about it, I would have been like, also, this article that you just published.
And we did write that with, and Leslie is someone who's a much more advanced isotope person than I am. So I love collaborating with her because she actually runs the instrumentation, and she's deeply embedded in all things isotopes. And she co-edited a book about food forensics and isotopes recently as well. So I love collaborating with her and others, but I think it's a very accessible article for people who are interested in kind of where this is going in forensic anthropology.
Mm-hmm. My second question is, what are you reading for work right now?
I like to read a lot, and I try to read about 50 books a year. And I try not to read books that are like just about, let's say, bone chemistry. I mean, I will read those things. Of course, I read lots of like journal articles, but I like just to read anything that's about science education, things like that. So I just like to, it's my distraction, I guess you could say. Instead of watching more TV, I like to read a lot. Currently, I'm reading The Fifth Beginning, What 6 Million Years of Human History Can Tell Us About Our Future by Robert Kelly. It's a short little book written by an archaeologist, but it piqued my interest, and so that's the one I'm currently reading right now. A recent book that I did read that is about isotopes, but it's more about radiocarbon dating, but it's called Hot Carbon. And that book is a fun book if you wanted to learn about the discovery of radiocarbon dating and the sort of applications to things like climate change. And you'll find that some of the earliest researchers dosed themselves pretty heavily with radiation in their research. And so they definitely took a hit for science.
Yeah. This is just a shout out for any science history books that you might come across. I always find them fascinating. Scientists are characters. I don't know if you know, I mean, obviously you personally, Dr. Bartelink, know scientists in real life, but listeners, if you don't know any scientists in real life, strong recommend. They are a hoot and a holler.
Absolutely.
And historically, even more so because people didn't know about stuff like radiation.
Not know that they were killing themselves, but yeah.
Yeah. Third question, final question, and then I will let you go. What are you reading for fun right now or consuming for fun? So it could be podcast, television, book, whatever it is.
So I mentioned I do read a lot, and I consider the reading the nonfiction fun, but I really like the Dark series. There's been two. It's a German Netflix series that involves time travel, and it is so mind-altering that I've had to watch each season several times because you pick up a lot of information. It's very confusing, and it's also subtitled or overdubbed. I'm lazy, so I went with the overdubbing, but it's a really cool show. I really enjoyed it, and it really made me think a lot. So that was definitely something that was quite fun to watch.
Well, what a great recommendation. Thank you so much. And thank
Absolutely.
you very much for joining me. I really appreciate your time.
Definitely. And thank you so much.
I bet you thought it was over. And it is. But I wanted to remind you one last time to send me those cannibalism expert recommendations. Just shoot me an email at gabby.lapera@anthrobiology.com. Have a wonderful day. Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show was produced and edited by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.